Okay, we're going to look at how to make a genomic library in this video. Now, in the previous video, we looked at how to incorporate bits of cDNA into bacterial plasmids. So, cDNA is DNA which is made from mRNA. So, mRNA in a cell, use that to make a template. Use that as a template to make a cDNA copy of it. And you then insert that DNA into these plasmids. Okay, so they see the bit of foreign DNA, which is like come from a human, made from the mRNA in a human cell or whatever cell, is copied, is put into this plasmid, and then when the E. coli divides, the plasmid also divides and makes multiple copies of this foreign bit of DNA, which you can then examine. Here, we're not going to use cDNA. We want to use the whole genome, usually like a human, the whole human genome. We want to chop it up into little bits. There's the whole genome. We're going to chop that up into lots of little tiny bits. And we want to stick them into something like a plasmid there. And so that we can grow the E. coli, get loads of copies of the plasmid, loads of copies of our foreign DNA. However, the plasmid is not really a suitable way of doing this. Okay, so the plasmid is called a vector. Vector means a carrier DNA, and it can't carry big enough bits of DNA, and it's not efficient enough to make this really practical for the massive size of the human genome, which is 3 times 10 to the 9 base pairs. It just can't cope with that. So we need a better vector. The vector we're going to use is this thing here, which is called, it's a virus which infects bacteria. It's called the bacteriophage, and it's called Phage Lambda. Phage Lambda is the bacteriophage which infects E. coli, which we can grow in very large numbers, and we know an awful lot about E. coli, know an awful lot about bacteriophage lambda, so it's a good way of doing it. So just to explain how bacteriophage, what its normal life cycle is. So this is a bacteriophage. It's got a cap protein, and it's got a tail protein, and the red bit inside is the bacteriophage DNA. It's its genome. So when the bacteriophage recognizes a protein on the surface of E. coli with its tail, it binds to it. Once it's bound, it then injects that bit of DNA into the E. coli. The bacteriophage DNA is actually linear. It's not like a plasmid circular, or E. coli. Chromosome is circular. That inserts into the E. coli chromosome, and it shuts down the normal functions of the cell. And instead, what it does, here we can see it makes the cell, takes over the cell apparatus in just making lots and lots of copies of this cap protein and of the tail protein, and lots of copies of the genome. Those and the phages are matured, so it's the DNA is packaged inside the cap, and when that happens, then the cell lyses, bursts, and all of these phage progeny come out. There's usually, I've drawn 5 of them. The usual number is about 200 progeny, which means offspring, per E. coli. So the strategy here is to, it's obviously a very good way of copying, making multiple copies of DNA. You start off with one virus up here, and at the bottom we end up with 200 copies of it, and those 200 can go and infect another 200 cells and so forth. So the idea is to get this viral DNA, there it is, chop a bit out and stick in a little bit of human DNA. And then that way you can make multiple copies. You can make as many copies of those little bits of human DNA as you like. So how does it work then? So you get some human tissue. It can be any tissue as long as it's got a nuclei in it with DNA. And you extract the DNA. And then you cut the DNA and digest it partially with a restriction enzyme. Now you may remember a restriction enzyme is an enzyme which cuts DNA at certain sequences. It recognizes a sequence. Usually it's 4 or 6 bases long. You'll recognize that sequence and it cuts it there. Now the reason why it's partial digestion is because if you allowed it to completely digest it, the bits of DNA you'd get would be too long. They'd only be about like 200 bases long, and you really want to get fragments which are about 20,000 base pairs long. That's the idea. And you want to insert them into the into the bacteriophage DNA. So just to imagine this is a piece of human DNA, and you won't just get, you're not just taking it from one cell. Obviously you've got this like tissue here, you've got millions of cells, so you're going to cut millions of bits of DNA off, and you end up with a series of overlapping fragments. So you may get some quite short ones, you get ones like that which overlap, and so on. So the idea is you will get coverage of the whole, the entirety of the 3 times 10 to the 9 base pairs of sequence. So those little bits of DNA, then first of all, they have to be, before you can insert it in the virus, you have to, so there's a bit of DNA, you have to put on them these green bits which are called sticky ends. And that means they do that so that they can be inserted into the into the viral DNA, which has got complementary sticky ends. We'll just explain what that is in a minute, but here's the viral DNA. And there are 2 restriction enzyme sites here. And the restriction enzyme is called E.coli. Okay, now all this bit of viral DNA here codes for non-essential things. So if you get rid of them, it's not going to disrupt the life cycle of the virus. It can still reproduce. So you're going to cut that bit out, and that gets thrown away. And then these are called the vector arms, these 2 bits, and you actually purify them on a polyacrylamide gel to get them away from the bit of virus DNA you don't want. And then you get your human bits of human. You mix it up with human DNA, and so you're going to get multiple viral vector arms with all those different bits of fragments of human DNA inserted into there. Just to explain how sticky ends work. So sticky ends, you get these whenever you cut with a restriction enzyme. So in the case of E.coli, you end up with the sticky end has got AATC. So what you get is, you're going to get there AATT. So that's this bit sticks out a little bit more, and when the other bit, which is kind of complementary to that, I'll do that in purple. That's going to have the overhanging bit that way, and the sequence there is going to be AATT. Okay, so when these 2 bits come together, what's going to happen is, here, I'll just draw that, it's going to go like that. All right, they're going to match. You're going to get base pairing in this section here. And then DNA ligase comes along, that's another enzyme, and just knits the 2 ends together. So you need to have your human fragments here, with the correct sticky ends to fit into where that end, where the sticky ends of the virus DNA there. So you've done that, and you've got all your bits of viral DNA with your human bit stuck in it. So here we've got you've got all of your millions of bits of, well, won't be quite millions, certainly hundreds of thousands of viral DNA with different bits of human DNA stuck in there, giving you coverage of the entire human genome. And that is then put into a beaker here, which contains the viral head protein, phage head protein, and the tail protein. You actually get these from a mutant strain of Phage Lambda, which, when the virus doesn't assemble properly, it makes all the bits, but it doesn't assemble. So you can get loads of head protein, loads of tail protein. You mix loads of bits of DNA in there, and what you end up with is you end up with your intact viral particles with a little bit of human DNA in there. Right, so, and you've got like maybe 100,000 different ones of these, and they're all different. Now you've got to reproduce those, and the way you do that is you get a Petri dish. There's a Petri dish, and on here you grow a lawn of E. coli. So you put enough E. coli on there, and you allow it to multiply so that it covers the entire surface of the Petri dish with a very thin layer of E. coli. Then you're going to get your viral, so you get all your mixture of your viruses, and you chuck it on there. And each one of those, when they go there, is they are going to cause an infection of an E. coli cell there. They're going to burst that. You're going to get 200 progeny. Those progeny, they're going to infect the ones around it, and so on and so on. You leave it for a few hours, and you end up with lots of tiny little clear areas there. And those clear areas are where you've got one phage, and each one's going to be different. You've got one phage with a little bit of human DNA in it, and it has caused lysis of these bacteria, and it's multiplied. So you've got multiple copies of your human DNA there, multiple copies. Right, now usually you can fit about, and you still see them okay, you can usually see 10,000 of these little plaques, these viral plaques on the surface of one dish. And that means you need about 15 to 20 Petri dishes to cover the whole human genome. And that is your library. It's like a little library, instead of books, it's a library of phages. And each phage has got a little different bit of human DNA in it. But the important thing is that you can do whatever you like with that. You can grow as much of that DNA as you want, and then you can, at a later date, you can sequence it, you can do whatever you like with it. Now, how you look at those is called screening, and that's going to be a different video because this video is going to be a bit too long. But what you can do is what is placed on here is you put a piece of nitrocellulose filter, and you blot it on the surface, and it transfers the DNA. It'll be a bit of viral DNA with the insert onto the filter, and then you can do what you like with it. Meanwhile, these Petri dishes can be stored in the cold, where nothing's going to happen to them. They're fairly stable, and you can find out which colonies are the ones which contain which bit of the human DNA, human genome, that you are particularly interested in. But that, as I say, we'll leave that for another video.